take care of that. So here we are. We're in chapter 4. It's about the cell. Most of what you will see from now till the end of this chapter, we've already discussed. You know, I've gone in and I've told you, what every structure is and what its function is, what it does. So we pretty much have done this chapter. I do want to just run through the PowerPoints, mostly to make sure that I'm not missing anything, because occasionally I do. And so it's just basically to check myself out, make sure everything's going okay. This is rough endoplasmic reticulum, what you're seeing here. I can back up. So if we go into the animal cell, I say too many times. I'm getting tired of it. I'm sure you're way sick of it. I'm tired of it. But anyway, this is the rough endoplasmic reticulum right here. You have the ribosomes attached with these little bitty dots. And that's where you put the amino acids together to build your proteins. That's what happens there. And that's why it has a rough appearance. And then, thank you. All right, so let's go to that other slide. I just wanted to make sure everybody understood where we were in the cell. It's always good to have an orientation. It's like, oh, where am I? I do that a lot. Like, where am I? where did I park? Where's my car? Is this my house? Does this key fit this door? You know, that kind of thing. Do I know anybody in this room? You know, that kind of thing. Should I be at this party? Probably not. Anyway, it's a crazy kind of existence. Yeah, it's a joke. I just try to have fun. I told you when we started, I like to have fun. Not everybody understands that type of humor. It's okay. I don't expect anyone to laugh, but I get a charge out of it. It helps me. It's about me, not always about you. But anyway, going to this, you can see that this is a ribosome that's attached to the endoplasmic reticulum. So this is a tube system. So it's our rough endoplasmic reticulum because we have a ribosome. And you have an RNA, and that M stands for messenger. Really, this is, if you'll remember the DNA, which is double-stranded, it will not get out of the nucleus. It cannot fit through the nuclear pores because it's just double-stranded. It's too big. It cannot get out. So what we have to do in order to get the genetic information out of the nucleus, we have to make a copy of that DNA molecule. So let's say, here's your DNA. It's a double-stranded molecule. We'll say it looks like that. 2 strands, and you've got, say, bases on here. Adenine, thymine, or thymine, adenine, cytosine, guanine, so forth. You've got all these nitrogen bases on that DNA. And the adenine always pairs with the thymine, and the cytosine always pairs with the guanine. But, you know, like I said, this is actually too big to get out of that nucleus. I wonder if that'd work. No, it doesn't. I tried. Again, orient ourselves a little bit of orientation. So the DNA is double-stranded. It won't fit through those nuclear pores. So what I have to do is, on the DNA double-stranded, I have to make a single-stranded RNA, and because it's just one strand of sugar phosphate base, you know, those are your nucleotide units called monomers, and you put those together to make a long single-stranded RNA. But it is small enough it can get through a nuclear pore and then therefore come out here into, there you go, brother, come out here into the cytoplasm. There, that's your messenger RNA, that red squiggle there. And it will associate with one of these ribosomes, and then that really carries the genetic information out of the nucleus. So you've got double-stranded DNA, has all of the genes, and I've said before, All that the DNA does is tell you what order to place the amino acids in. So do you put number 7, bond it to number 3, bond that one to number 20, bond that one to number 18? You only have 20 different amino acids. What sequence do you put those in? So when it folds up, you end up with a transport protein or a membrane protein that can move something in and out of a cell, or a pigment. We've talked about different things, enzymes. So that is the function of the DNA. It just carries the genes. Personally, it doesn't do anything. It doesn't do anything. It just sits there. But the RNA, we have to actually get that out of the nucleus because it's a mirror image of one side of the DNA. DNA is double-stranded. We're just going to make a mirror image of one half of that. And it is single-stranded. That can get out, the RNA, it can get out of the nucleus and associate with a ribosome. And then that's where you put the amino acids in order. So really you're carrying the genetic information out of the nucleus by way of that RNA molecule, and then you're putting that together. So this is really what they're showing you here. Okay, so I have the ribosome, and you have the RNA that associates with the ribosome. A ribosome is really a two-part unit. You have a smaller unit, as you can see, and then you have a larger unit. And that little ribbon that's running through there, that's the RNA. So that's a genetic code that used to be on the DNA. It's a mirror image of one side of the DNA molecule. And, you know, so it's going to tell you what order to put those amino acids in. So that's what this actually is. You have amino acid number 3 next to number 3, next to number 12, next to number 14, next to number 2, next to number 17, next to number blah, blah, blah. So that's your first amino acid, second amino acid, third, fourth, fifth, sixth, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, 36, 37, 38. Once we get to 50 amino acids in length, got ourselves a protein. That's why they're calling this a polypeptide. We don't have 50 yet. So it's a shorter chain of amino acids. So it's still growing, seems to be. They're still adding amino acids. So as you go along this, you read this information that you have on the RNA, and that tells you what order. The first amino acid, the second, which ones you're going to put in order. And 20 next to 3, next to 12, next to whatever. And build protein. And then once you've built it, it will automatically fold into a three-dimensional shape, which is what they've done. They're showing you that it is, at this point, you know, basically carrying this shape. And it may or may not have sugars attached to it. I put a plus or minus. May have those, may not have those. Doesn't have to. It just could be all protein, and that would be it. All amino acids that are folded into that position. And then if you want to move it somewhere, you create a bud. It's like it buds off. So the membrane begins to kind of pull to the interior, and then it continues to pull to the interior, and then eventually, you know, you've created that, which is what I refer to as a vesicle. That's what that is. It's just a membrane sac. And then you can move it. And you can move it, say, like, to the edge of the cell and export. If you want to export it out of the cell, maybe you want to take it to the Golgi apparatus. You want to combine it with some other things. So let's look at that Golgi again, just to make sure everybody understands how this is all working together as a functional unit, because it's important. You know, I mean, these cells have to stay alive. In order to stay alive, there are certain things they have to do. So, you know, maybe what you're doing in that example, instead of exporting something, you've got a little bag. This one's coming from Smooth ER. They don't really have one showing coming from the Rough ER, but we could have another little bag with protein in it, and then it goes into the Golgi apparatus, and maybe the sugar or the lipid comes from the smoothie arc. That might go into the Golgi apparatus, and you repackage it together, and then you end up producing another bud, and this thing right here, which actually it would be this one, would be our vesicle. So that you might want to export out of the cell, depending on what the cell needs. Some things they export. Your salivary glands make an enzyme called amylase. Anything that ends in an ASE in biology is going to be an enzyme. So you have like amylase and protease and carbohydrases and lipases and, sucrase and lactase and maltase, and the list goes on. A bunch of ACEs. ASE tells you it's an enzyme. So, you want to, let's say this is a cell that's in your salivary gland. It's a glandular cell, and you want to secrete salivary amylase. And so then that will eventually move over and then attach to the plasma membrane. And then once it does that, then you're going to export whatever is inside of this thing. So it attaches, and then, this enzyme is exported, whatever that enzyme may look like. That's how I typically draw them, right there. So you have just exported an enzyme, and that goes actually into a tube, which drains into your oral cavity, and then you have saliva. And then you swallow it, and then you make some more. It doesn't take long before you swallow some more. I mean, those salivary glands are constantly making that, and then you're swallowing it, and they constantly make that. Just a constant, a constant event where you're sending sugars and you're sending proteins and sugars into the Golgi, packaging that together, and then mailing it out of the cell, which it then goes into a tube, and then that's where it goes to the oral cavity. Is this always going on? And pretty cool, just to have some understanding of how that actually works. And maybe you don't want to export it. Maybe whatever this thing is, this little vesicle, maybe it's something that you want to build. Maybe the cell is getting bigger and bigger, and it wants to divide into two cells. Happens all the time. Cells divide to produce two cells, which they divide to produce four, and those divide to produce 8. Cell division is happening. It's like red blood cells. They only live for about 100 to 120 days. That's it. So they're dying off all the time. Your liver is where they're broken down. And if you've ever eaten liver, it has kind of a weird flavor to it, like liver and onions. My mom used to feed us that occasionally. I was like, oh, it's liver and onion day. This is a bad day. Suck day. Liver and onions. Boo. But it has that weird flavor because that's where red blood cells were broken. There tends to be a lot of iron in that. So it's like biting into a nail, kind of. It's got a iron flavor. Not really tasty. Anyway. But it also has a lot of sugar. Remember, we store sugar to sugar to sugar to sugar in the liver. So if we eat too much sugar, then we store it in the liver. So liver and onions has a sweet flavor to it, but it's got that funky iron in there that gives it a crazy flavor. But it's kind of sweet too. So, and of course the onion has a lot of sugar. We've already been doing that in the lab. So, liver and onions has a lot of sweet flavor to it. And then if you're cooking it like in butter, which is the way I would cook it, cook everything in butter. As long as I'm taking my combination of vitamins, then I'm good. I can cook with butter. I'm sorry. Yeah, it can fold up into a three-dimensional shape there, which is really what they show. on that other slide. That's what they're showing you, that it can be, you can add things to it, you can take things off of it, that protein, and so really on that slide, a pretty complex little event going on. On this slide, what they're showing you, of course, is you're making the sequence of amino acids, and then it folds into this glycoprotein, and then we're exporting it, or basically putting it somewhere. If this is mucus in the nose, then we're exporting it into the nasal cavity. But yeah, it goes through a folding activity, and like I say, that is maybe somewhere where you may want to add things to it or take some things off of it. So modifications can be made before you use it, before you export it out of the cell, or before you use it inside the cell. So you're doing lots of combinations, even within the endoplasmic reticulum. You can do more combinations once you send it to the Golgi apparatus, but there are things that occur to whatever you built, the protein. Again, you can attach sugars to it, you can attach different things, you can take little pieces of the amino acids off of it. Once you've made that sequence, maybe you want to remove something. Maybe it doesn't really need that entire sequence, but every time you remove something, if I remove this little sequence right here of 3 amino acids, and I bond 1 amino acid at this end with an amino acid at that end, I should make that a little longer, and I took this little section completely out, I've made a completely different protein. It will be a different protein altogether, and if I take this section out here, remove that, I've made a different protein. Maybe I want to add a little section in. Maybe I want to attach sugars to it. Maybe you do all kinds of different modifications to that molecule that you produced. So even once it's manufactured, people think, well, okay, it's static. It's just manufactured. You're done with it. You're finished. It's over. You don't mess with it. You continue to mess with it. Absolutely. And that happens in the endoplasmic reticulum. It happens in the Golgi apparatus. So yeah, you do all kinds of modifications, which is interesting. That a cell, one cell, can do all of those things. And you start thinking about all the cells that make up the pancreas. And some of them are doing this, and some of them are doing this other thing, and some of them are making this thing, and some of them are making this other thing. Some of them are exporting this, some of them are exporting that. And that's just the pancreas. Well, then you have the liver, which is huge, the largest internal organ of your body. Same thing. Different cells are doing different activities within that one organ. The brain, different areas of the brain process different areas, different bits of information. Depends on where the information ends up. Does it end up over here? Does it end up over here? Does it end up over here on the brain? So, and that requires different activities to occur within that cell. So, to simplistically put this, like what we're doing right here, that's a very simplistic way to look at it. I mean, it's really a major operation, and very interesting once you begin to understand how that really does work. And I don't think you can ever really understand it completely. I think it's so complex, it's really very difficult to do that. And you just try to wrap your mind around as much as you can, and you'll take other classes and they'll continue to talk about these things, and eventually little pieces start to fall into place. And at some point, you just kind of go, you feel like, and I don't think you can ever really get there, but you feel like you have a very good understanding of the way that cell actually operates. Now, and like I said, I don't think you can ever really get there to have a full understanding of the way it operates. It's very difficult to get to that point. There's so much that goes on, and that's just with, again, one cell. So, of quite the amazing little structure. You know, and the cell is the smallest unit of life. I mean, that's the smallest thing you can have that would be alive, would be that cell. Okay, this is going through things that we've talked about. Smooth-ER does a number of things. One is produce lipids. All of these other things are lipids, oils, phospholipids, steroids. They're just lipids. I don't know why they said lipids, comma, oils, which are lipids, comma, phospholipids, which are lipids, comma, steroids, which are lipids. They could have just put lipids and left it alone. Or if they wanted to put lipids, they should have put such as oils, phospholipids, and steroids. That would have been better, actually. They didn't. Okay. And this is, you probably looked at how many authors you have on this book. I think there are like 5 different authors. And the book is really coming mostly from a Campbell book. And Campbell was probably the best writer for biology books that we've ever had. And he's passed away. So, and even he made mistakes. I could go through his Majors book, which is, if you think this one is maybe a little difficult to read, if you got the Majors book, this is a mixed majors, meaning you could use it for a majors class and a non-majors class. But if you got the Majors book, you would go, oh my God, it's tough. It's tough. Even for me to read it, I would go through it and I'd have to read maybe some paragraphs 2 or 3 times before I would go, okay, I get it. All right, and then move on to the next paragraph. And I'm thinking students do not need this book because it's that difficult to read, even for someone who teaches it and has taught it for so many years. So I shied. We used to use it. I shied away from that book. It's like, no, I'm not making them read this thing. Okay, also, and I did mention this, processed drugs, like alcohol, or drugs and alcohol. I guess they separated those. Alcohol, I suppose, is a drug, I would say. Oh, you got enzymes that'll break down antibiotics, enzymes that'll break down THC, enzymes that'll break down cocaine, enzymes that'll break down, whatever you happen to, put in your body. So there are many, many enzymes there that can handle what they say the harmful substances. And actually, when it comes to weed, they're now starting to breed it, and many of you probably already know this. There are really 2 major molecules that you have in marijuana, weed, which would be, one is a THC and the other one I tend to forget. It's a C something. Anybody remember that? C what? CBD. Okay. Well, that ought to be pretty easy to remember. CBD. But CBD is like a painkiller. And they have also discovered that CBD will stop people from having seizures. So there are people who have seizures. I have a real good friend, and he comes from, he's a Hurt, if you know the Hurt family in Odessa, Texas. They're the original Ratliff Ranch, Ratliff golf course. They have just humongous land holdings and oil holdings and real estate holdings and so forth. So, and he started having seizures. And in fact, he and his wife were staying at my house one time in Alpine, and he went into a seizure. And so, his wife came and knocked on the door and said, Jay's in having a seizure, and you kind of have to hold them down or they could hurt themselves. And so, anyway, we went and helped him through that, and then we got him upstairs, and we were drinking coffee. This was about 4 o'clock in the morning. And then he went into a second seizure, and I turned to Vicki, his wife, and said, has he ever had 2 seizures in a row? And she said, no. So I went straight to the phone and dialed 911. And I was on the phone with this young man, And I said, giving him the address, and we need an ambulance here ASAP. And while I was on the phone with the man, he died. His heart stopped. He died. And I was talking to the young man on the phone. I said, he just died. I said, we're going to try some CPR. We're going to try to see if we can bring him back, but I need to go, And so the ambulance showed up. We did get him back, and on the way to the hospital, he had a third seizure. He'd never had anything like that before. So when I found out that this CBD was something that they could breed now, where I don't think Jay, although I don't really know, it wouldn't surprise me if he smoked weed. It doesn't really matter. I don't really care. It's not anything significant to me personally, but whether someone does or doesn't. But when I saw the special program about that, I just texted him and I said, Jay, have you heard about this compound? And they're now breeding it in very high concentrations in marijuana and breeding the THC in very low concentrations. So you could smoke this, or you could get a distillate of it and take that, and it stops you from having seizures. I mean, he's like totaled 3 pickups. 3 pickups. Just drive along, go into a seizure. You can't control anything. He's going to kill somebody. He's going to kill himself. And, I was like, man, you need to get a hold of this stuff for sure. Anyway, so there are beneficial effects to that, absolutely. And then the THC, it just is, makes someone a little, give someone a little buzz, maybe different than alcohol. But I would certainly ban alcohol completely, and I'd certainly vote for marijuana to be legalized. Get rid of alcohol. That's dangerous. The marijuana, I don't see that as a danger, and there are benefits to it also. I don't see any benefits to the alcohol at all. None. Anywhere. All states. Sure. Okay, cool. I'll text him about that. He may know about that by now because he texted back and he said, funny you should say that. I just got another text from my daughter who's, anyway, she's in Austin and graduated and, 30-something years old anyway. And she had told him about the same thing, that she had seen. Bless you. Do you do those just alone or, I mean, it's more than one? Oh, okay. All right. Well, that's good. Okay, I understand. I'm allergic to molds too, which is what that is. It's a mold spore. Anytime it rains, it's mold spore, so I'm stopped up. Absolutely. And I did take a antihistamine this morning because I knew I'd be like, and I am. Anyway, so that's the smoothie R. I did kind of go off task a little bit there, but I think some of that information is very beneficial, especially if you know someone who does have seizures. This kind of information is critical. If you have it and you can put it out there for people, I think it's great. You may help save someone's life or save someone in an automobile driving go through a seizure and kill a whole family or something. I mean, it's good to pass on good information that can help people. Okay, anyway, rough endoplasmic reticulum. What you're doing is, for the most part, you're making protein and transporting that. That's really what's happening there. This right here, it shows you the cross-section of the Golgi apparatus, and this, of course, is an electron micrograph superimposed on just a diagram of that is what you're doing. So we're sending something into the Golgi apparatus, and then it's moving from these are what they call saccules. So what we might think of as pancakes all stacked on top of one another, that they call saccules. And then it just moves from one cycle to another, to another, to another, and you're putting things on it or taking things off of this molecule, whatever it is, or this combination of molecules, and then you're transporting it. Again, you may use whatever that is inside the cell. You may transport it outside the cell. So that's the Golgi. The lysosome, we did mention, contains digestive enzymes. It can digest worn-out cells. If you have an entire cell that becomes non-functional, and it begins to happen. Your cells begin to just die off at a faster rate than they're produced, and that is the aging process. So if a cell were to die, you don't want to keep it in the body, so you just want to digest the whole cell. So lysosomes are released in large numbers so that it's called an autolysis, so the whole cell is just destroyed. The cell produces the lysosomes that destroy the entire cell, which is very cool. And then you could also have the destruction of the organelles that wear out. We did mention that. We did talk about trying to grab a lizard's tail, and the neurons that are sent to an area of the tail all release lysosomes, so the tail falls off right there. If you engulf something into the cell, you may want to destroy whatever's being engulfed. Basically, it's food. White blood cells just engulf anything that shouldn't be in the body, so that happens a lot. So. Food vacuoles, digest food, destroy bacteria, fuse with other vesicles containing damaged organelles. We talked about all that. And this will give us an animation that will not work. But this gives us something. We're actually engulfing something. In this case, it's a solid material. If you engulf something into the cell and take in a bulk amount of it, a large amount of it, not just molecules moving across cells. So this is what they call a bulk transport, B-U-L-K, a bulk transport. Whatever this stuff happens to be, I have no idea what that is. It doesn't really matter. But, we're introducing this into the cell, this material. And it's a solid material, more or less. It's not a liquid. And so when you're doing that, that's called a phagocytosis. It's actually a cell eating. And then you destroy it. Cytosis means it's a destruction. The cell destroys it. Well, the lysosome destroys it. That's where your digestive enzymes are. So a phago or a phago, that would be to engulf is what you're doing. So you're bringing it to the interior. And like I say, that can be any number of things. If it's a liquid, this is for solid material. So this is just for solids. If it's for liquids, you call it a pinocytosis. So you can also engulf liquid material, like if you have a sugar solution that you really want to quickly pull in because sugar's good, you need to use it, but you wouldn't want to destroy it. You just want to pull it into the cell and store it, the sugar. So you can engulf the liquid, the sugar solution. So that's a pinocytosis, so that is for liquids. Another good example of that would be removing ammonia from the blood. The kidneys do that. They can pull in a whole bunch of ammonia very quickly. Digest, that's mostly for the solids material. You don't want to typically digest the liquid materials. You might carry that over to the lysosome where you can detox it, or the smoothie R where you can detox it. Anyway, you can do any number. You can do many things with it if it's a liquid material. Okay, so it's kind of giving us an idea how that's going to work. So we're pulling something in, and then once it completely buds from the plasma membrane, you're really just making that plasma membrane a little bit smaller because you're wrapping it in plasma membrane. So the membrane gets a little bit smaller, and they call that a food vacuole. And then The lysosome will fuse with it. You eventually digest it, and those particles are released. Here you have a old organelle that's not functioning or not functioning well. This would be a mitochondrion. So you can actually fuse with something like that and destroy it and get rid of it. And that's pretty cool. Again, we don't have our video. For some reason isn't working. Here are some other vacuoles that we haven't talked about. This is a contractile vacuole. These are used by your aquatic organisms, and it sort of looks like this, and then you've got these long structures that go out around it like that. And this thing will actually have, and that continues all the way around like that. But anyway, you have a pore in the center of this contractile vacuole. So in the case of a paramecium, they might feed on a smaller organism and engulf it, but when they do that, they're also pulling in water. So the excess water that comes into this cell needs to somehow be exported out. So you pull it in using these long membranous leg-like looking things. You pull that water into there, and then it goes into this contractile vacuole, this thing right here, and then from there, you can export it. It kind of contracts, and really you're squirting water out of that bag. So it's to eliminate excess water. That's how that works. It's interesting. A lot of your aquatic animals have those, or organisms will have those. Typically one-celled organisms will have those, not a larger creature. That's not going to happen. Okay, so here's a central vacuole. We've talked about that in plants. You can see the chloroplast. This is an electron micrograph. You really don't see the cell wall. It's too small to be seen with the electron micrograph. Okay, well, we've kind of discussed that. Golgi apparatus makes lysosomes with digestive enzymes. Maybe we send a protein from a rough endoplasmic reticulum into the Golgi, and then we can send a fat or a sugar from the smooth endoplasmic reticulum. They both go in to the Golgi apparatus, and then they move from one vesicle to the next vesicle to the next vesicle, and eventually then they will bud off of that. Well, that doesn't look great. Bud off of that, and then of course you may export it, or you could use it within the cell. I mean, they always show an export for some reason, but you don't have to. Maybe something you need within that cell, but it's not a graduate level course, so I probably spend a little more time than I need to talking about these things. But it doesn't hurt anything. Won't kill you. I don't usually talk about peroxisomes. Now, mitochondria and chloroplasts, notice evolved by what's called an endosymbiosis. Now, if you break the word down, like endo means to bring to the interior. So this would be to bring in. Again, it's Latin, to bring in. A symbiosis, this is an association of two different species. You have lots of symbiotic relationships. You have a bacterium in your intestine called E. coli. Also, the full name is Escherichia coli, E. coli. And E. coli and you get along really well. He's in your intestines. We feed him. We eat food. And a lot of that will go eventually into the intestines. And so he gets a little bit of it. And he goes, that's good. All I have to do is hang out down here and get fed. Of course, he has to hang out with the poo, so I don't, if there's a second life, I don't think I'd want to come back as an E. coli. Like, yeah, God, could I go as an E. coli next? That'd be great. Yeah, live in the intestines, super. Nothing I'd want to do better than just live down there with all that poo. No, not anything I really want. So we give them food and water. And then what they do in exchange for that, the E. coli provides vitamin K. Bless you, vitamin K. And the vitamin K helps our blood to clot. So if you cut yourself, hopefully, unless it's a severe cut, your blood will clot. And you should, anytime you have a wound, you need to have some blood loss. People are like, oh my goodness, I've got a cut. I need to stop the bleeding. Well, the bleeding is part of it's part of the process. You want it to bleed some, because when it bleeds, some of whatever you've introduced, so if you punctured yourself, whatever was on that, you have bacteria, dust particles, pollen, spores, who knows, something on it. Tomato protein, if you're cutting a tomato and accidentally cut yourself a little bit, you don't want tomato protein floating around through your body. You're not a tomato. All right, look in the mirror. You're not a tomato. Get a tomato, get side by side, look at it, look at you, check it out. You are not a tomato. So, you don't want the tomato protein. So with the bleeding, you're actually allowing those things to come out. That's a good process. And people are like, oh my goodness, I'm bleeding. This is not good. I'm like, unless it's a gusher. Every time your heart contracts like that, you've got blood going, shooting out, for six feet distance. That's not a good situation. You better get better get something tied around that real fast. But other than that, let that puppy bleed a little bit. That's good. You want that. Not bad. And it'll clot on its own, unless it's something severe enough that you're going to have stitches. So if you have to go to the hospital, then at that point, no, it is not going to clot. It's just too big an insult to your skin and to your body. Bless you. That it isn't going to clot. So you have to get stitches. I don't know if I've ever had stitches or not. I don't really remember. Isn't that weird? You get so old, you're like, have I had stitches? I don't think I have. Maybe I have. So it's an association of two different species. It's like E. coli provides vitamin K for us for blood clotting, and we provide food for it. So, we are mutually benefiting from that association. Very good thing. All right. So what is thought to have occurred was that at some point in history, and they'll show us a diagram of this here shortly. At one point in history, mitochondria are approximately very near the same size as a bacterium. So, and there's a lot of support to this. Mitochondria only have one circular DNA. Bacteria only have one circular DNA. Okay? Mitochondria have 70S ribosomes. Well, bacteria only have 70S ribosomes. Out in our cells, because we're eukaryotic, we've got true nucleus. Our ribosomes are bigger. They're called 80S. They're just bigger. It's a totally different type of ribosome. It's different. So, and then some other things, the mitochondria and the chloroplast, they're double membrane. So let's say that you have a cell and it engulfs another cell. Okay, so it does, and then that pinches off. Well, I have the membrane of the cell. This is actually plasma membrane there. And then the membrane of the bacterium would be this second membrane that's more internal. So this would be, say, like bacterial membrane. So you would get two membranes. Also, if you had something that produced something as important as ATP, why would you want to produce a lysosome and fuse with it and destroy it? It's producing energy for you. What would be the point? So at some point in history, we go from having just prokaryotic cells to having eukaryotic cells, and that was like 1.3 billion years ago. The first eukaryote was discovered in the geologic record at 1.3 billion years ago, although there weren't any people back then, but we can radioactively date these things. And so the idea is they used to be kind of bacteria. And they were bacteria that were engulfed and they were not destroyed. So mitochondria and chloroplasts. You have bacteria that are also photosynthetic. So if we engulfed one of those, we would end up with a chloroplast. So the photosynthetic bacteria, most of those, not all, photosynthetic bacteria, we call those cyanobacteria. Cyanobacteria. Some people refer to them as blue-green bacteria. They were called blue-green algae back in my day, but turns out they're actually prokaryotic, so they're a bacterial form, but they are photosynthetic. So if you were to engulf one of those, then you created a chloroplast. You've got a double envelope structure that has one circular DNA, 70 S ribosomes, double membrane. And by the way, the mitochondria and the chloroplasts, they can divide independently of the cell. Now, not outside the cell, but within the cell, they can divide. So you can have, say, one mitochondrion that divides into two, and they divide into four. So if all of a sudden I start lifting weights, which someday hopefully I will, my cells are going of juice, and they shouldn't. They should be certainly good for at least an hour, and way beyond that. Although I will tell you, these recorders, they really drain the energy. So, and let me just real quickly show you how that is believed to have occurred. It's a real simple thing. It's called endosymbiotic or endosymbiosis. It's a theory, which means it's not just a hypothesis. It's a theory, which is the last step you have before you go into a scientific law, which in biology we call a principle. We don't use law in biology. We use principle because I think people would go, wait, we have a principle of evolution instead of the law of evolution. I'm afraid Texas would just, shoot all the biologists, hang us. We'd all be swinging from trees if we could find a tree in Odessa that wasn't dead. Has to be. We have to have a few. We just have to hang a bunch of people on one tree. It'd be kind of like a, ghoulish, Halloween's coming up. That would be kind of a interesting Halloween decoration for a tree. So, again, what you can see here, we have a structure that is engulfed, and let's say this was a bacterium, was a prokaryote, and it was engulfed into this cell. Oh, do that. Forget that. Give that up. Okay. So, the prokaryote, oh God, I did it again. I'm a moron. Here we go. We're there. We're there. All right, so the prokaryote, which would be a bacterium, was engulfed. And then this thing started producing ATP, the mitochondrion manufactured energy, ATP. So instead of producing a lysosome that would fuse with it and destroy it, we didn't do that. And then if this is a blue-green photosynthetic prokaryote, which would be a cyanobacterium, you know, if it were engulfed, then it would develop into a chloroplast. And it would have two membranes, bless you. Two membranes are about the same size as a bacterium. They can divide independently of the cell as long as they're in the cell. They can't outside the cell. But so there is a tremendous amount of support to the idea that they used to be independent organisms, prokaryotes that were engulfed, and now they live inside the eukaryotic cell, which is pretty cool. Nope. All right, we'll pick back up there. 43, we're about finished with this chapter. So that's good. Don't save any weird things that I did.